Hi everyone, my name is Katie. I teach chemistry and biology at Science Leadership Academy at Beeber, and I'm excited to tell you about what we did during these past couple of months with Sandra, Dave, and Cauleen. And thank you, Vicki, for organizing all of us. Also, we were the cancer biology seminar, and we learned a lot. So we talked about what is cancer, how is cancer diagnosed, the evolution of cancer. We talked about cancer in other organisms besides humans. We talked about immunotherapy, how we can use our immune cells to genetically engineer them to target cancerous cells, which I didn't know was a therapy that was developed in Philadelphia. Cancer treatment, we had a really interesting discussion about social determinants of health, several discussions about that. We talked about Henrietta Lacks, a woman born in the 20th century who was the unwilling source of the first human line of cells that was really crucial for research. We also had a really cool opportunity to actually work with some model organisms. So we all had packages of fruit flies delivered to our houses. I actually got two packages of fruit flies because the first box got lost and then showed up on my porch like a month later. So now I have twice as many fruit flies that I would ever really need, but I'm excited because some of them are actually still alive and I can use them. We learned how to grow fruit flies. We learned how to study them. We learned how to observe them under a microscope. We actually got microscopes, which is really helpful for me as a teacher, like always in need of new supplies. These fruit flies that we experimented with were genetically engineered to be predisposed to have cancer. So it was cool to see the difference between the normal type and the cancerous type. My curriculum unit is titled Delicious and Nutritious Chemicals, an idea for engaging 10th grade chemistry students in the study of biological molecules. I'm envisioning a 4-week unit where my chemistry students will get to experience the same things that I experienced in the seminar, actually handling these model organisms, as scary as it might be. It's really interesting to see in real life what an experiment can look like, especially with something like fruit flies, which I know in the past I've experimented with my students with bacteria, but fruit flies are way more exciting than bacteria. So in the first week, we would talk about the molecules that keep us alive: carbohydrates, proteins, lipids, and nucleic acids, which is like pretty standard high school biology stuff. But we would take that a step further and talk about how these macromolecules actually sustain life and what would happen if we feed fruit flies exclusively these molecules. So I'm envisioning that I would train my students to handle the fruit flies using the techniques that I learned in seminar. And then we would have students choose a macromolecule. So for example, if students should choose carbohydrates, maybe they would feed their fruit flies like pure cane sugar and then see what happens. And it would culminate in a week of presentations where students can have the same experience that I'm having right now with presenting their learning, sharing with their classmates. And I really just wanted to create a unit that would have an emphasis on lab skills. And thanks to TIP, I now have access to materials to do that. So I'm grateful for that. So yeah, thank you.